Hey guys, welcome to part 1 of cell structure or cytology. So the cell is the basic living structure and functional unit of the body. It's a living structural and functional unit of the body. Like remember we said physiology focuses mostly on the tissues and the cell. Any processes, structures mostly rely on that. And functional, of course, cells make up tissue make up organ, and that is the structure of the body. It's our anatomy. So the definition is basic living structural and functional unit of the body. There are 3 cell theories. Number 1, the cell is the fundamental unit of structure and function in living things. All living things are made up of one or more cells. Then cells arrive from other cells through cell division. We have 2 types of cells. There are prokaryotic cells. These are cells whose organelles are not bound by cell membrane, for example, bacteria and archaea. Number 2 is eukaryotic cell. These are cells whose organelles are bound by membranes and have membrane-bound organelles like plants, animal cells. The cell is comprised of individual components with specific function essential to carry out life processes, and that includes the cell wall, cell membrane, cytoplasm, nucleus, and cell organelles. And we can see in this structure you have the cell plasma. This is the structure of a eukaryotic cell. Have the cell plasma membrane. We have the mitochondria, that is double layered, that's double bilayer membrane. We have the intermediate filaments, one of the cytoskeleton. We have the ribosomes that are bound on the rough endoplasmic reticulum. Some are just on the surface according to their function. Rough endoplasmic reticulum arising from the envelope, nucleus envelope. Then the smooth endoplasmic reticulum are on the lateral sides. Lysosomes, we have the excretal vesicles. Peroxisomes, we have the vacuoles. We have this Golgi apparatus with the Golgi vesicles, and the cytoplasm, that is where the organisms mostly are found. In this nucleus, we have the nucleus envelope, we have the nucleus itself, nucleolus, we have the chromatin materials, and the nuclear envelope has pores, as we can see it here. Cytoskeleton include microtubules, microfilaments, and intermediate filaments. Okay, so let's discuss about cell membrane. Cell membranes, plasma membranes, surrounds the cytoplasm of a cell. Mostly it's made up of a phospholipid bilayer with embedded protein, carbohydrates, and cholesterol. The phospholipid form 2 layers with the hydrophilic heads. So the heads are hydrophilic. They attract water, they love water, soluble in water, I can say like that. So they're facing the watery environments, and the hydrophobic, they are the tails now. So the 2 layers, the heads are mostly in the either extra or intercellular fluid, then the tails are inside each other. So proteins are interspersed throughout the layer, some spanning membrane integral, like through the membrane, others attached on the surfaces, like either peripheral side or inside the cell. Cholesterol molecules are scattered within the bilayer to maintain fluidity. Semi-permeability, bilayer controls in and out movement of substances. Transport protein facilitate diffusion, active transport. Also glycoproteins and also glycolipids help in cell recognition and communication. Main functions of the cell membrane is to separate the cytoplasm inside a cell from extracellular fluid, separate the cell from one another. They provide an abundant surface on which chemical reaction can occur from the structures, receptors, and other things. They also regulate passage of materials in and out of the cell. Also let some tissues in and keeps others out. They qualify as selective permeability. That is the cell membranes. Too much, but that is in a nutshell. We can also discuss the cytoplasm, which is a thick like substance found inside the cell membrane. It's made up of two main parts, cytosol and organelles. The cytosol is the fluid portion containing dissolved nutrients, ions, and also enzymes. Then the organelles are specialized structures found suspended within the cytosol, mostly to provide medium for chemical reaction. Cytoplasm also allows the diffusion of materials throughout the cell, supports and protects the organelle. We can discuss the cell organelles. We can start with the membranous organelle that have membranes. Number one is the nucleus, which is large membrane-bound organelle found in eukaryotic cells, usually located at the center of the cell, contains the nuclear envelope, as I said, nucleoplasm, chromatin, and other nucleolus. It also contains the genetic material, that is the DNA, to form in the form of a chromatin, just dispersed like that. The nuclear envelope is double membrane that separates the nucleus from the cytoplasm. Nucleoplasm or nucleus is the jelly-like substances that suspend the chromosome and the nucleolus. Nucleolus is dense, dark, stained region inside the nucleus and main function to make ribosome, which is the cell, helping cell synthesize cell protein synthesis. Nucleus stores genetic information, also controls cell activity such as growth, metabolism, production, and regulates protein synthesis by controlling transcription of DNA and okay, so we can talk number two. That was number one of the membranous organelle, that was a nucleus. We can discuss mitochondria. These are made up of two membranes. So the inner and the outer membrane, they enclose the matrix containing enzymes, ribosome, and the mitochondrial DNA. The outer membrane is smooth, alafu it's permeable to small molecules, while the inner membrane is highly folded into cresti that increase surface area for reactions to take place. Mitochondria are the site for aerobic respirations and produce energy for cell in form of ATP, hence enabling cell activities during muscle contraction and other things. Lysosome is also membrane bound, and there are small organelles found in the cytoplasm of an animal cell. Each lysosome is enclosed by a single membrane, contains a powerful hydrolytic digestive it's a digestive enzyme. The single lipid membrane keeps these enzymes separate from the cytoplasm, preventing the damage to the rest of the cells. Lysosomes are formed by budding from the Golgi apparatus, which packages the enzymes into vesicles and so forth. So the main functions of the lysosomes is to break down old organelles and in the process of autophagy, worn out cells, and also foreign substances like bacteria. Another organelle that's membrane bound is Golgi apparatus. A Golgi apparatus is a membrane bound organelle found in the cytoplasm of a eukaryotic cell, often located near the endoplasmic reticulum and the nucleus, made up of series of flattened membrane bound sacs called cisternae, along with small vesicles used for transporting the proteins packaged and so forth. So it receives vesicles containing newly synthesized protein and lipids from rough and smooth endoplasmic reticulum, and it sends out modified materials, secretory vesicles, to their final destination, maybe the plasma membrane, lysosome, or secretion outside the cell. The enzymes within the cisternae modify, then they sort and package the protein molecules that are brought in. To secrete a substance, small sacs of the Golgi membrane break off and fuse with the cell membrane, releasing substances to the exterior of the cell in a process called exocytosis. So in a nutshell, Golgi apparatus help in modifying proteins and lipids. They also, for example, like during carbohydrate process to form glycoproteins and glycolipids, also sorts and packages molecules into vesicles. They produce lysosome, play a role in secretion and cell membrane renewal. That was a Golgi apparatus. Number 5, in the membrane-bound organelles, we have endoplasmic reticulum. Endoplasmic reticulum is the network of membranes tubules, and flattened sac cystae found throughout the cytoplasm of eukaryotic cells, but it's divided into two according to its function and structure. We have rough, covered with ribosomes. Smooth doesn't have ribosomes. So rough endoplasmic reticulum synthesizes and transports protein. Mostly those secretions are for use in the membranes and so forth. For the smooth, it synthesizes lipids and steroids, detoxifies drugs, and stores calcium ion, especially in the muscle cells. Both types help form a transport vesicle that moves substances to the Golgi apparatus. Let's discuss about transport vesicles. Are small membrane-bound sacs found throughout the cytoplasm of a eukaryotic cell. They move materials between organelles such as the endoplasmic reticulum to the Golgi apparatus. From the Golgi apparatus to other destination, like we say, the cell plasma membrane. Each vesicle, remember, is bounded by phospholipid bilayer membrane that encloses a fluid-filled interior containing proteins, lipids, and other molecules being transported. Vesicles bud off from the membrane of an organelle, like for example interplasmic reticulum or Golgi apparatus. So they are moved through the cytoplasm by motor proteins such as kinesin and dynein that travel along cytoskeletal microtubules. When they reach their target organelle or the cell membrane, they fuse with it, releasing their content. We can discuss also peroxisome, which are small membrane-bound organelles found in the cytoplasm of a eukaryotic cell, especially mostly in the liver, kidney, for detoxifying. Each peroxisome is closed by single membrane and contains oxidative enzyme, mainly the oxidases and the catalases. This keeps oxidative reactions separate from isocytoplasm. So oxidases mostly catalyze reaction that produce hydrogen peroxides as a byproduct, and the catalyzes immediately breaks it down into water and oxygen to prevent cellular damage. That is the most important component of it. Want to know that peroxisome lack DNA and ribosome. The enzymes are synthesized in the cytoplasm and imported into the organelle. They can grow and divide to form new peroxisome in the process. And this means peroxisome help mostly in breaking down fatty acids by beta oxidation. They also detoxify harmful substances such as alcohol and other toxins, and maintain redox balance within the cell. In some cells, they also assist in lipid synthesis such as cholesterol and plasmalogens imported in brain and heart tissues. Those are there are 5: the membrane-bound organelles, nucleus, ribosome, Golgi apparatus, peroxisomes, and rough and smooth endoplasmic reticulum. Let's discuss the non-membranous components. Number 1, of course, is ribosomes, which are small and bound membrane organelles found in all cells. They are made of all cell, we mean prokaryotic and eukaryotic. So they are made of ribosomal RNA and protein forming 2 subunits. They may be free in the cytoplasm attached to the rough endoplasmic reticulum. So we have those that are free, and we have those that are attached on the rough endoplasmic reticulum. They are said for protein synthesis, that is reading mRNA and joining amino acids into polypeptides. Free ribosomes produce protein for use inside the cell, the ones that are not attached on the ear. So the ones that are attached on the ear make protein for secretion or for membranes inside the cell membrane or outside the cell, extracellular side. But those that are free in the cytoplasm, they make protein to be used inside the cell. Okay. Number 2, non-membranous bound organelle is the microtubule, which is a hollow tube-like protein structure that is found throughout the cytoplasm of a eukaryotic cell, forming part of the cytoskeleton. These microtubules maintain cell shape, support organelle movement, and form the spindle fibers during cell division. These spindle fibers help in separating the chromosomes and so forth. So they also make up the core of cilia and flagella, which are used for cell movement. That means we need to discuss flagella. Flagella are long, white-like extensions found on the surface of some prokaryotic and eukaryotic cell. In the eukaryotic cell, we have sperm cells. Flagella generate cell membrane, cell movement. They wipe or rotate to propel the cell throughout the liquid. That's why the sperm is able to move from the vagina to the fallopian tube. But that is the only cell that has a flagella in the eukaryotic cell, mostly human body. They are powered by ATP, eukaryotes mostly, and proton gradient in the bacteria, that is the prokaryotes. This enables locomotion and sensing the movement. So flagella are motor appendages that allow cells to move and respond to their surrounding. We have another cell, which is organelle, which is a cilia. Cilia, they are short and have hair-like extension found on the surface of some eukaryotic cells. Mostly in the respiratory tract, we have them that waft out pathogens and other things. We have them in the fallopian tube to waft the egg. So these beat coordinated waves help in moving substances across the cells, such as mucus and even the egg. We can also discuss other cytoskeletons. In the name of cytoskeleton, we said it's a group of fibrous proteins that provide structural support for cells. So we said it's good for motility, cell production, transportation of substance. Cell production is cell division, transportation of substances within the cell, and make up of 3 types of protein-based filament. We have microtubule, the one we've just discussed, and intermediate filaments and microfilaments, which are actin filaments sometimes. So microfilaments are the thinnest components of cytoskeleton, composed of actin protein subunits arranged in a double helical chain. They are concentrated just beneath the cell membrane, where they maintain cell shape, enable cell movement. So together with myosin, they bring about muscle contraction, as we discussed in the tissues. Assist in cytokinase, splitting of cells into 2 during cell division. Intermediate filaments, they are rope-like structures, but they have different proteins. We can have keratin, vermentin, or lamellins. The most stable actin filaments provide mechanical strength, helping withstand tensions, forces, and other things. So they anchor organisms like the nucleus, and in epithelial cells they form a desmosomes, junctions, that connect adjacent cells for tissue integrity. Can finally discuss the microtubules we discussed, but I can just pass through it. Under centrioles, the thickest of the 3 is microtubule, and structural filament components of subunit protein called tubulin. Microtubules maintain cell shape, structure, help cell compression of the cell, help resist compression of the cell, and also play a role in positioning the organelles within the cell. Microtubules also make up 2 cellular appendages that we discussed. Centrioles found in animal cells are cylindrical structures made of 9 triplets of microtubules. A pair of centrioles form the centrosome, which organize microtubules during cell division, as we mentioned earlier. We can discuss cytoplasm and cytosol, which is cytoplasm includes, of course, the cytosol and organelles. And for cytosol, is the gel-like liquid that surrounds the organelles. It's mostly water. We said it contains water, ions, enzymes, and nutrients. It's a site for metabolic pathways such as glycolysis, and helps transport materials within the cell. Hey, let's see each other in our next part 2 section of the cytology. We will be discussing cell division. We'll be discussing different DNAs, division and other replication and protein synthesis. So see each other.